Hi, everyone. Welcome to the Career Roundtable. Today, we are going to be joined by Irv Weissman, Shaheen Sikander, and Wolf Wiedemeyer. The way this is going to work is we're going to have them introduce themselves. They'll talk a little bit about their career trajectory. We'll go through some questions and then for the first half hour, and then we will open it up to audience questions. So during the first half hour, you can submit your questions in the Zoom chat. After the first half hour is done, we'll go through the questions, and then I will call on you to turn on your camera and microphone and ask your question live. So with that, we will go ahead and welcome our panelists. Great. Irv, do you want to go ahead and start us off?
Sure. Well, obviously, you can look at me. I'm not at the beginning of my career. I started in science. When I was 10 years older, I read a book called Microbe Hunters. And the main point about that, these are the people who discovered what microbes were, and then they did clinical translation of their discovery. And I think that set my career in place. By the time I was 16, I was working in a lab, small lab in Montana. I decided medical school is the way I wanted to go, but I only applied to one medical school, Stanford, which at that time had a 5-year rather than 4-year medical school. You spent half of your time, the first 3 years, half of every day was free to do research or scholarship. So I, since then, have been dividing my time between academic and research. I've spent my whole career at Stanford. I'm now head of the Stem Cell Institute. I used to be head of the Cancer Center. Before that, I was head of the immunology program. The main thing that drove me is the science I followed, not academic ambitions. I'm a perfect example of what they call the Peter Principle, that is, promote people beyond their expertise. That's my story.
Great, thank you. Shaheen, would you like to go next?
Sure. So I am just starting out my independent career. So just started my lab about 6 months ago at UC Santa Cruz. And so ever since a kid, I wanted to be a scientist. I did not know what kind of scientist, but just the fact that I wanted to be a scientist, and my main goal was to find life outside planet Earth. That was my goal, because I thought that that's going to change everything once we find life. And as I got more interested, I learned more and went through high school. I really was very enthralled until today is how cells divide and how they maintain their states. And so, I come from India, so I did all my education, undergraduate education in India. And when you come to high school, when you finish 12th grade, and if you're good in science, you have two choices. You become an engineer or you become a doctor. And when I told my parents I didn't want to do either of them, they were quite disappointed. And I said, I'm going to just follow basic scientists and science and do a PhD. They are not in science themselves. So for them, it was a little hard, but they were very supportive. And then I applied to PhD programs in the US, and I was fortunate to be accepted in a few, and I did my PhD at UC Irving. And over there, while I was studying DNA repair, at that time, cancer stem cells were just identified in Michael Clark's group and then subsequently other groups. So after my PhD, I decided to join Michael Clark's group at the Institute of Stem Cell and Regenerative Medicine at Stanford. So the same building that Irv helped build, or actually spearheaded in building. And then I did my postdoc there. I did a slightly longer postdoc and stayed on for a little bit more because I wanted to have that opportunity to explore independent science. And when it came to deciding what is the best path forward, after a lot of exploration, I wanted to stay in academia primarily because, as I said, I'm still fascinated about how cells maintain themselves, how they decide to divide and how they decide not to divide. What are the signals? And being in that basic biology is how I ended up being at UC Santa Cruz. A second thing was also the fact that I was inspired by my teachers, so I wanted to be in education and especially in undergraduate education. So that's my story.
Great, thank you. And Wolf, would you like to tell us about your career trajectory?
Sure, yeah. So I always knew growing up that I wanted to be a biologist. I think and that's very similar to Irv and Shaheen. I think, field biology is more what I was envisioning back then. So I got my undergrad degree in zoology in Germany. But then when I was looking for a PhD, I also wanted it to be relevant and concrete in a sense. So I decided to go to the German Cancer Research Center, and I joined Manfred Schwab's lab, who was working on NMIC at the time, and also started working on the emerging field of apoptosis regulated cell death. So in that sense, I've kind of come full circle working for AbbVie now, who are one of the leaders in the field of targeting apoptosis or therapeutically. So did my PhD in Manfred's lab, and then for a postdoc, went to Boston, Dana-Farber, the Cancer Institute in Boston, and really became interested in mouse modeling, so utilizing mouse models to model human cancers. But at the same time, functional genomics, and that was around the same time that the Cancer Genome Atlas project started. So we did a similar study in our lab where we profiled human glioblastoma tumors. And that kind of was similar to what the TCGA pilot project then did. So like around that time, I became really interested in patterns, so genomic patterns that could inform therapeutic choices, as well as pathways, and specifically cell cycle pathways. So was very fortunate to be able to work with palbociclib, the CDK4/6 inhibitor, before it became this blockbuster drug, and was really fascinating to see some of the resistance mechanisms that continued treatment with this inhibitor elicited in cancer cells. I then, when it came to kind of making the next career step, I considered both academia and industry at that time. So for personal reasons, I wanted to relocate to the West Coast. Being in Boston, I wanted to go to Southern California. But I interviewed at the time with both Pfizer in La Jolla, as well as Cedars-Sinai in Los Angeles, and decided to go the academic route at that time. And hindsight is 2020. I don't know if that was a good decision in retrospect, but it was definitely an interesting choice. And so I had my own lab as an assistant professor at Cedars-Sinai, UCLA for about 6-7 years. I never got the big grants, so at some point, it was time for me to kind of decide what did I want to do, and I think being able to do relevant, meaningful science, I wasn't able to do that in where I was in my career at the time. So then decided to make the move to industry. And since Los Angeles does not have a strong industry presence, it's kind of emerging, but didn't have it at the time. Then relocated to the Bay Area and joined AbbVie about 4 years ago, and really haven't looked back since. I'm really enjoying it, and it's a move that was overdue for me, I think. That's it.
All right, thank you. So kind of building off of that, for our trainees who are looking at industry versus academia, what are things that make good industry scientists versus academic scientists and researchers, and how did you know that that path was right for you? So Irving, let's move back over to you. How did you know that academia was the right path for you, and what makes a good scientist in that field?
Well, when I started, we all thought that the pharmaceutical companies were evil. So it was an easy choice. But as my career went on and biotech started out, in 1981, I was on the founding scientific advisory board for both Amgen and DNAX. I learned a lot from that. I learned that I didn't want to actually work in industry, but I liked advising, and I liked the chance that you could help direct a project and make it more likely to succeed. But I also knew that today's discoveries become next year's or 10 years down the line therapies. And if you gave up doing discovery research, you would pretty soon not be able to bring the insights of discovery research to it. So on my own, I decided the way I was going to translate our discoveries was either like Amgen or DNAX, participate, but in 1988, we discovered the mouse blood-forming stem cell. And I had a number of people who urged me to form a company. We formed one called Systemics, where we isolated the human blood-forming stem cell. We invented, actually back at Stanford, a mouse that had the human blood-forming and immune system organs. We had to obtain them from human fetal remains. So we had to immediately, while we were getting in there, work on the ethics. How could you obtain human fetal remains without encouraging people to have abortions, without altering the best of health care for the woman who's decided irrevocably to have an abortion? So we worked out those things so that nobody could make a profit. Neither the woman nor the doctor would sign on that they were going to donate the tissues. It's just after the decision is made and at the time of the operation or the intervention, we could make sure that nobody had the motivation of ending a pregnancy so that we could do research or they could make money. So we eliminated all of that, and we did that by 1988. We were able to show that if you put in the human fetal bone for bone marrow, human fetal liver, which is also blood forming, human fetal thymus to make T cells, and human fetal spleen and lymph nodes to receive all of those cells to make an immune response. And we showed if we put all but the bone under the kidney capsule, they not only rapidly were vascularized, but they interacted. So we could show, and we were the only ones who could show, we could make a human immune response. And we could inject HIV and get AIDS in the mice. Back then, none of you even know this. Robert Gallo and Luc Montagnier were battling it out about who discovered HIV. They both had in vitro CD4 T cells, which they cloned out or grew HIV on it. And this is the important point, the reason I'm taking so long on this. Those viruses that they had cloned, sequenced, and claimed firsts on didn't cause HIV. if you grow HIV in vitro, with each replication, if you're not carrying the properties that make HIV pathogenic, you lose them just by random variation. And the retrovirus is not absolutely perfect in making an exact copy of the RNA. So we had to go to patient samples. And we received patient samples, cloned the virus out of them. They were always pathogenic in this mouse. They always got rid, eventually, of the peripheral CD4 T cells. And we even found out why AIDS is so bad, because once it gets to the CD4 T cells, the early progenitors within the thymus that give rise to both CD4 and CD8 T cells, you wipe out the capacity to make new T cells. Now, I'm giving you a lot of time on that simply to say that shortcuts don't work. Don't go in vitro if you can do it in vivo, because only the body is organized to do things that we don't yet understand. So ever since I've been in science, I've followed the mantra, in vivo veritas. If you want to know the truth, go work in vivo. It's harder. I'll just say that the second part is that when our group, and then with Mike Clark's group, identified isolated cancer stem cells or leukemia stem cells, so that was, ours was 2000, the big companies would come to us and say, that's such a great thing. Boy, can you give me some cancer stem cells? I said, can you work in the body of a mouse that has a human primary tumor in the right organ? Do you have the capability of isolate the normal stem cells from the organ to compare to the cancer stem cells? No, they said, but we have cancer stem cells already in culture. I said, no, you don't. You have cells in culture that are so long in culture that they aren't close to the body. So you will end up studying something which has a high chance of being an artifact. And if you like to spend your life following artifacts so that you can make money, go ahead and do it. But we're going to keep working in these hard systems to find out what's going on. So my advice to people is that you can form companies, or you can go to companies that are formed. But look out at what kind of science you're doing. And if you are serious about science driving you and your own thoughts driving the science, you probably ought to look at academia first. Because even the companies I've founded, and there's a large number of them now, Systemics, Stem Cells Inc., Celerant, 47 Inc., and so on. Even if you're a founder, in the end, the money makes the decisions. And when the money makes the decisions, you have to be happy working for a group where it is not just your science that makes the decision, but where the business people who are on the board think the money comes from. And I've always marveled that in the beginning, they trust you if you've made a discovery, because they're kind of interested about how you made the discovery. But somewhere after a year or two, they all think that they understand the field as well as you do. And then they start making scientific decisions. And if you're lucky, they made the right scientific decision. If you're unlucky, then they'll just abandon it and go on to the next one. So I know that's a long answer to the question. I decided to stay in academia to make discoveries. I decided to learn how to help form and then advise and then eventually be directors of companies I found, mainly to try to keep the decisions relevant to and dependent upon scientific discoveries.
Excellent. Thank you for sharing all of that and the background. So Wolf, you already talked a little bit about switching over from academia to industry. Can you talk a little bit more about why you made that switch and why that was right for you, and maybe what makes a good industry scientist?
Sure. I feel like I have to defend industry a little bit more. So what I've found is that, both in academia and in industry, I think we're both scientists, or most people that I've met are scientists first. I mean, there's passion here as well about doing good science, advancing the right programs, and bringing the right kind of medicine to the right people at the right time, right? For me, it was, I like working on something that is concrete, in that I think it's also a personality question, right? I'm interested in a lot of things. I like hearing about a lot of different things, and being in industry gives me the opportunity to work on different project teams, to kind of see how much actually goes into making a drug, and how difficult it is to bring a drug to patients. That is something that I wasn't really aware of, or fully aware of when I was in academia, I think. The other thing I like about industry is that it is a team effort, just because, like I said, it takes so much to get an IND filed and to bring a drug to patients, that you have to work as a team, and it's a great opportunity to work with other functions, that if you have your own lab, and you're kind of used to doing everything yourself, which is also great, of course. And I fully agree. I grew up thinking that pharma companies are evil, of course. We all have these notions, but I feel like I found that at the end of the day, we're all scientists. We're interested in the science, and we want to bring the best product forward.
Great, thank you. And then Shaheen, I know you're starting out relatively recently compared to Irv, for example. And you talked a little bit about why you wanted to stay in academia, but could you talk a little bit more about how you knew that was the right path for you to stay on?
Yeah, so I'll be honest, I explored extensively both career paths. I mean, being at Stanford, there are startups happening. And so you invariably get involved in discussions. And so I explored it. I even had, I work closely in advising 2 companies. And I was, but at the same time, I just felt like having the ability to sort of just discover something new, and like looking, as I said, I'm always intrigued by these cells and how stem cells are choosing to divide versus not divide it. And I just felt that there is more to be discovered. And there are a lot of constraints that I had to think of also. I don't know, it all happened at the same time. I was considering industry, but I wanted to apply to academia first and, make sure. And it just so happened that it worked out for me. Also, I have a family, so I probably wouldn't have moved somewhere far away to start my own lab. The fact that I had this opportunity at UC Santa Cruz was very important. I have continued talking and advising these companies that work with patient-derived xenografts, because that's my expertise. So I do meet with them and tell them, you should design your experiment this way and not that way. And I completely agree with Irv when he said, do that in vivo experiment, because it's harder, it's longer, it's more painful, but it will give you the right answer. And so it's nice. And as Wolf said, and on the other end, when I'm talking to people from the industry and designing, it's fun to work with them. I see them work as a team, and they come we can work together. So, but one other important thing for me was to be really involved in education and training. And UC Santa Cruz, allowed me to do that. So I'm involved in teaching cancer biology, and I can talk to them about the experiments that I'm doing in lab and how it's relevant to them, and the excitement that I see in those students. It's kind of amazing. I've just started my lab, and we have 3 undergraduates who came before. They have not had any previous lab experience. They are generally not from, their parents are not from backgrounds that are in industry. And that is so rewarding to have them learn the techniques and then ask those questions. And it's amazing. They don't have the entire breadth of knowledge, but sometimes they ask a question and they're like, yeah, we actually don't know that, and we don't have the techniques to answer that. So it's very rewarding to see that. Yeah.
Great, thank you. All right, I'm going to ask one more question before we open it up to the audience. So now is a good time. If you have any questions, submit them in the chat. We'll do this one last question, and then we'll turn it over to audience questions. So what is the best piece of advice that you wish you had followed but didn't? Does anyone want to start?
I can start on that one. I think I think maybe see the writing on the wall. Like make sure that the working environment that you choose is right for what you want to do, and that you have the necessary resources to do it, right? I think, and again, I don't think industry and academia in that sense are too different. I mean, they're great working environments. I remember working at Dana-Farber in the kind of the Harvard community. It was amazing, right? The kind of the exposure that you got to world-class science. But then here in the Bay Area, in the industry setting, it's somewhat similar. You talk to other people from other companies. There's so much innovation going on here. So I'm inclined to say, make sure that whatever you choose, it's a positive, stimulating, energetic, and supportive working environment.
Thank you. All right, Shaheen or Irv, would either one of you like to go next? Best piece of advice you were given but didn't follow, but wish you would have?
I didn't follow advice very much. People told me when I finished medical school and decided not to do internship and residency that I was making the mistake of my life. But I knew that if I were going to understand my field and the adjacent fields I needed to understand, that I couldn't do that and also maintain the knowledge base to run a clinical practice of some sort or to teach clinically especially. So I decided that I'd take the risk. And I would say at about the time of my tenure, I wondered if I'd made the right choice because there were a lot of rumblings they didn't want to give me tenure. But then magically it did happen anyway. And so I could say, well, I was right all the time. And for me, I was right because I love to know the adjacent fields. And I want to clarify one thing about being an industry scientist. I'm not against it. Obviously, many people who train with me end up in industry. I was trying to give you the caution. But now let's think about being an academic scientist, starting off like Shaheen. You've got to organize the lab for yourself. You have to attract the people. You have to write 2 or 3 grants to get one, at least. You have to deal with the crap when somebody sends you back, they reject your grant, and you realize by what they write, they didn't understand what you were doing in the first place. So you take on a lot to become a lab leader. The field you're in is competitive, and people who are in the field grew up in competitive environments. Competition doesn't always breed honesty. So you can see some of your data, and even if you have an inkling that it might not really be true in vivo, you'll still publish it. And you get a little further out on the limb. Now you have to defend what you published. Amy Wagers, Camilla Forsberg, and I tested many of the assumptions when they said blood-forming stem cells turn into brain, blood-forming stem cells turn into muscle, turn into liver. We tested all of them, and none of them were repeatable. In industry, it has to be repeatable. And I find there's more general honesty in industry labs because you know you're going to be checked, you know you're going to have to prove it to the FDA, and you know pretty soon people's lives depend on it. The people in academia who allow a shading of the truth to drive their ambition, I believe will eventually be caught out. A guy who went from academics to industry at Amgen, I'm not going to mention his name, looked at papers in Nature, Cell, and Science and looked at their reproducibility. Did they lead to people who could even repeat the studies? And only 30 to 40% of them were reproducible. Now you have to ask yourself, why does that happen? Well, you got to get a Nature, Cell, or Science paper, every postdoc thinks, to get an academic position or a good industry position. Those three are actually for-profit entities, even though science claims it isn't. Look at all the ads. So they have to attract a bigger audience who will read the ads, and that shades their editorial decisions. So I would just say that you will always be testing your own integrity in an academic position and in an industrial position. In the industrial or biopharma positions, it's more likely that the people checking and trying to repeat your work will find out what wasn't really true. But you can go a long distance, unfortunately, in academia and make a reputation. But the bigger they are, the harder they fall.
All right, thank you, Irv. I will say that you're not taking advice sounds very serendipitous. And thank you for offering the opposite perspective of going into academia. And then Shaheen, will have you conclude with the best piece of advice that you were given and didn't take, but wish you would have.
So my piece of advice is a very practical one. So when I was starting off my postdoc, somebody said I should learn how to code, and I didn't. So I think like if you are getting in, starting your careers, I think the best thing is a story where basically there are two woodcutters. They go every day and they chop wood from 8 a.m. to 5 p.m. And one of the woodcutters chops wood the whole day. And the other one takes a break every day for an hour. And after many years, the woodcutter that takes a break ends up getting more wood. So finally, this other woodcutter asks the first woodcutters, how is it that you end up always chopping more wood, even though you take a break? And the other woodcutter tells him that's because in that 1 hour, I'm sharpening my axe. And I think that's the advice is sometimes you have to go out and learn new techniques and that are outside your field to sharpen that axe. And that's one thing that in coding is one of those things that I did not do early on. I'm doing it now, but so just take that time to sharpen your axe every so often.